when you're under stress, your evolutionary history is that you might get torn apart, and you really need to have a potent inflammatory anti-pathogen response to that pending event. Well, you've hit upon one of the fundamental challenges that we as a company and as innovators are facing, which is it's a platform. What we've built, what we've invented is a true platform. What we've invented is a way to take out the soluble target and not affect the membrane target at all. That's a huge gap in the existing pharmacopoeia. That's not just a smart question. It's almost like you were eavesdropping on a conversation we had with our team this morning. So there is a debate internally in our group as to whether...
Lou Hawthorne is the founder and CEO of NaNotics and the inventor of the core technology. He has 27 years of experience founding and leading companies developing new technologies in biotechnology and life sciences, including reproductive cloning, human IVF, oncology, and artificial intelligence. In 2002, he was one of the first people to develop a cloning technology company called Genetic Savings and Clone in BioArts International, which was later acquired. There's a lot that we get into in reference to NaNots. This is like really the core of what we get into today, but we also get into his history, cloning, what's it like working with high net worth individuals. Lou is a true innovator in the aging, longevity, biotechnology space. This episode captures just a small thimble of what all he is up to, all that he has done, all that he has accomplished. Learn with Lou is really where serendipity happens, because you never really know when knowledge is going to come in handy in your life. And what Lou's working on is truly inspiring, as well as the fact that he's not a PhD scientist, and yet he's a self-taught autodidact figuring something out that's going to impact millions and millions of people. There's a lesson out there. There's so many listeners, so many of you writing, commenting, asking, how do I get in? I have this type of degree, or I have no degree, or I have this type of passion. How do I get where I want to be going? I think this episode really is a great testament to the powers of being an autodidact, being completely ruthlessly, not ruthless in a bad way, but like just so focused and passionate about what you're working on that you want to dedicate your life to it like Lou has. I want to thank Lou again for coming on the show. I want to thank every single one of you, wherever you are, for listening in and tuning in and being such avid supporters of the show. Let's stay curious and learn about Lou on this episode of Learn with Lou. I think this is in the lifespan article you sent me, quoting PTSD, depression, and all sorts of cognitive disorders. These are also cytokine disorders as well. And so I would love to just start there and unpack that, because I've never thought of them as cytokine disorders.
Sure. So I want to just be upfront that knowledge is infinite, and everything we know is just a clearing in a forest, an infinite forest. So there's certain, I don't know if that's really an environmentally sound metaphor, but anyway, I've only cleared a certain area of knowledge. And I've done a lot more work in understanding the molecular roots of cancer and certain inflammatory disorders, including the most acute, which is sepsis. So our work does overlap with cognitive disorders, and here's how we view it. So anytime you're under stress, I'll take a step back and say that in our work, we study specific diseases, but we also study evolutionary biology, because everything that exists in terms of health and disease has its roots in evolutionary biology. And anytime we're under stress, there's a molecular response to that goes all the way back to when we were in danger of being ripped apart by predators. And so the stress response that we have in a challenging situation at work or with our significant others or family, that stress response evolved to deal with tigers. So it tends to be a really exaggerated response geared toward pathogen control. So the risk when you're under stress, your evolutionary history is that you might get torn apart, and you really need to have a potent inflammatory anti-pathogen response to that pending event. So what happens is the body releases these proteins that are toxic to pathogens. And normally they're delivered in a very precise focal way against a pathogen once it's in your body. But when you're under stress and your body doesn't yet know what that pathogen is going to be, it tends to release these things systemically. And it's known what they are. They're TNF alpha, IL-1 beta, IL-6. They're specific cytokines. And what all a cytokine is the signal molecule of the immune system. So immune cells communicate with each other and with non-immune cells via these signal molecules. And one of the messages that these signal molecules can deliver is a suicide message. It tells an infected cell or an early cancer to kill itself. Now, the problem is when you have a stress response and these cytokines are elevated and there's no specific pathogen, well, they cruise right across. First of all, they damage whatever tissue they touch, including healthy tissue, and they cruise right across the blood-brain barrier. Blood-brain barrier has evolved to filter out pathogens, but it doesn't filter out your own endogenous natural molecules. So these very potent cytotoxic molecules cruise right across the blood-brain barrier and wreak havoc in delicate brain tissue. And that leads to a variety of negative cognitive responses. There's pretty good evidence that plaque formation, for instance, in Alzheimer's is a protective response to elevated cytokines, including TNF alpha in particular. And also that certain situations like PTSD are responses to the trauma, the psychic trauma of combat. So even if the soldier isn't wounded, the wound can happen at a cognitive level based on this rush of inflammatory cytokines designed to protect against a wound that might not actually occur. So that's some of what we're interested in. Now, we aren't developing nanots to target cytokines to treat these cognitive or neurological disorders just because it's a lot harder to do that than to target specific diseases like arthritis, rheumatoid like rheumatoid arthritis or psoriasis, these well-characterized inflammatory diseases. But the thing that's cool is the targets are the same. So our work is focused on developing nanots, which are nanoparticles that absorb pathogenic molecules from blood. And this is at the molecular roots of disease. So it can treat specific diseases, or they can be applied to future applications like the ones you mentioned. Mm-hmm. That was a lot. Did that make sense?
It does. The interesting thing is that PTSD, like you can see it when they do MRI scans. And I've often wondered, what about our brains when we're exposed to something would respond to actually alter the way our brains look when we do MRI scans, like how the heat levels, all these different things. Like you can actually see the effects, even if there's no even if you didn't get bumped on the head or anything like that. Just being in an environment that causes you to have some form of PTSD, your brain will literally change. And I've often wondered, what's the process for doing that? Can you step in and counteract it? Is it the cytokine things that you've been talking about, or something else? Because I really feel for the veterans that come back, or people in everyday life that are getting PTSD, and how can we help them? I looked at psychedelics and stuff. Is that something that breaks it up? I don't know, but the underlying science of it's very fascinating to me.
Well, these cytokines are incredibly powerful. They're a given cytokine molecule is 1 million times smaller than a cell, but a single one or a small set of them can have a profound effect on a cell, either driving inflammation or cell death or proliferation. So it's not a surprise that a change in the signal molecules that pass between cells can have an aggregate effect at the tissue level that you pick up via MRI. So trauma and stress that shows a physical manifestation in a whole brain MRI, that's not really that surprising. It makes perfect sense. It's aligned with the molecular processes that are underway during stress.
Yeah. So eventually, we were talking maybe 10, 15 years down the path, because you already have a ton going on right now on deck in terms of what you're developing. You could see an application for your technology in mental health disorders like PTSD and depression.
Yeah, and I don't think it'll be that long. I mean, we've already been talking to DARPA and DoD about potential uses of our technology for warriors under stress or undergoing trauma. And some of this is physical trauma, because sepsis, which is an exaggerated immune response to a trigger of some kind, which could be a viral trigger or could be a wound, sepsis actually kills more people than all forms of cancer combined. And it's a huge concern for warriors. Sepsis on the battlefield, you can have a survivable wound and yet die of sepsis. That's really common. And in fact, it's the number one cause of death in hospitals. People go in for survivable conditions and they have an immune system overreaction and they die of sepsis. So that's probably our near-term use, is to develop nanots for treating sepsis, and then that would almost certainly end up being repurposed for dealing with PTSD.
Well, I saw in the list. I wish I could throw it up there for both of us to reference, but there was stuff from there from Alzheimer's to breast cancer, in terms of the different applicability of what you guys are working on. I think there was, if memory serves, you have mice models already proven out the breast cancer intervention for, and there was one other one that was related to the brain, I think, that you also had a mice model kind of proving it out, but I remember off the top of my head. Which goes to the question of, when you have all these different targets that you potentially could be hitting, how do you focus down into one? Is the sepsis the focus because it's the biggest, it hits the most people, it's the most acute in terms of response, that in terms of the research you've already done so far? Like, how do you, the big thing with learning or trying something, or when you have something that has potential, is like, how do you focus it down so it's not like all over the place?
Well, you've hit upon one of the fundamental challenges that we as a company and as innovators are facing, which is, it's a platform. What we've built, what we've invented, is a true platform. It's currently the only way that you can deplete soluble, meaning circulating in blood, soluble targets without affecting membrane forms of the same target. You could do it by pheresis, where you filter the blood externally, but there's nothing injectable. Pheresis isn't really scalable, but there's nothing injectable that can clear a given target that's soluble, but not affect the membrane equivalent of that target. I'll take a step back and explain why that's important. So in vertebrate biology, including human physiology, there's many targets that exist in both a soluble form, circulating in blood, and a membrane form. So the same target is on the surface of a cell of some kind. And it's also very common that the soluble form is pathogenic, is driving some sort of disease or a disorder of some kind. But the membrane form is doing something important. Now, drugs rely on chemistry, on biochemistry. So they are going to hit both targets. The targets are biochemically identical. What we've invented is a way to take out the soluble target and not affect the membrane target at all. That's a huge gap in the existing pharmacopoeia, drug toolkit. Drugs can't do that in general. And every nanot does that. So then we look at, to answer your question, how do we prioritize? Well, the first way we prioritize is we look at diseases where there is a soluble target that we want to take out and a biochemically identical membrane target that we don't want to disturb. Now, fortunately, almost every disease has that initial qualification. Then we have to look at, what is the unmet need? I mean, we both want to reduce suffering in the world, and we have to return value to our investors. We got to do both. So we have to look at the unmet need. Where are the drugs really falling short? In which diseases? And that will tell us where are we most likely to reduce suffering and generate value for our investors. Got to do both. Mm-hmm. So that further winnows our targeting. And then we look at feasibility. Like, which diseases are simpler than other diseases? And then we also look at which diseases have single targets, where you take out that one target and you're going to make a profound difference in the progression of the disease, versus multiple targets. Now, sometimes these align and sometimes they don't align. For instance, no tumor can survive without immune inhibitors. Tumors are grossly abnormal. And the fundamental question for the last 100 years has been, how can a tumor survive in a hostile immunologic environment? It's grossly abnormal. The immune system should clear them out as soon as they're formed. And the answer is, they protect themselves with immune inhibitors, just like a fetus. They use the same, it's not just analogy, they use the same molecules as a fetus to protect themselves. So, but we know that the tumor must comprehensively inhibit the immune system, and it uses multiple inhibitors to do that. You don't have to comprehensively disinhibit the tumor's defense in order to have a profound effect on the tumor. You just need to poke one hole. So you take out one of those immune inhibitors, and the immune system is then going to inflict major damage on that tumor without any drugs or radiation or anything. We've proven that in animals, and we've also seen predicate approaches using pheresis that have had efficacy far exceeding any drug. So just taking something out of blood that a tumor depends upon can have a profound effect. Now, sepsis, in some ways, is a relatively simple disease because we know, or disorder, we know the molecules that drive the cytokine storm that characterizes sepsis. It's well characterized, but there's no way to get them out. However, you're not going to be able to save most patients' lives by taking out just one of those cytokines. We think the minimum viable set of nanots to have a significant impact on sepsis, which again kills 11 million people per year, is probably 3. So here's a disease with huge unmet need that's relatively simple in its etiology, its molecular basis, but you're going to need 3 nanots. So that we have to drop that down a little bit in our priority. We're not ready to tackle that. We are ready to make a difference in cancer. So we're leading with cancer, and we're moving into inflammatory diseases where there's a single target driving that disease. And ultimately, that entire toolkit can be combined in different ways to treat sepsis.
So this comes to a head that I think kind of speaks to my ignorance. So for the longest time, when I pictured cancers being able to, hide in plain sight, I've always thought of it more like they wore a cloak, or like the inhibiting functions on them. I thought it was more like something they would have on them. But it sounds more like if you can take something out of the blood, that it's more like they emit the chemicals that more like a bath, like they kind of create an environment around them, but they can't be seen. So it sounds like it's more like the second than the first, or at least like a combination, and shows like my ignorance of the science of this. But I always thought it more like you had to find them, and then you had to bind onto the site, and then you attack them that way. But if you're able to just filter the blood out from the chemicals that are hiding them, and then they're able to be seen, it kind of seems like it's more the latter. Like they're not like having. If that makes sense, I feel like I'm talking a little bit out of like not very well.
Actually, what you're saying is pretty much aligned with current dogma on how cancer works. Even most serious cancer researchers view cancer's defense as being primarily a membrane-mediated defense. So you have molecules on the membrane of a cancer cell that inhibit incoming immune attack. That's kind of dogma. It's not correct, though, or it's not sufficient. It's not complete. So we actually know there is literature. There's substantial research, even though it's not yet dogma. There's substantial research that shows that tumors use a combination of inhibitors. They use membrane inhibitors, like right on the surface of the cancer cell, but they shroud themselves with a gradient of soluble inhibitors. So it's most dense in the tumor microenvironment, less dense in blood surrounding the tumor, and less dense still in general circulation. So this is actually analogous. I use a lot of military analogies because I really feel like in cancer, it's a war. It's a war between the tumor and the immune system. So the analogy that's most relevant here, we call it the bullets and bayonets analogy. Any soldier would prefer to use bullets before bayonet, because by the time you have to use a bayonet, the enemy's right on top of you. It's a last-ditch defense. Likewise, in evolutionary biology, the evolution of these membrane immune inhibitors is the last line of defense. And again, cancer just hijacks methods used by the fetus or the placenta. So the placenta has membrane immune inhibitors as its last line of defense. But before those get engaged, the last thing the fetus wants is to defend itself against immune cells that are right on top of it. It wants to neutralize the mother's immune system at a distance, but only in a boundary region. If the fetus neutralized the mother's immune system totally, systemically, the mother would die of whatever infections are floating around. So evolutionary biology had to come up with an arbitration between two incompatible survival advantages, one being having an active immune system, and the other being carrying young internally, right? Those are two great survival advantages. They both came up a couple 100 million years ago, but they're not compatible with each other, because if you're carrying young internally and you have an active immune system, those are incompatible. So you need some sort of way of arbitrating between the two. And what evolution came up with was local immune inhibition in the region right around the placenta, with a mix of soluble immune inhibitors to neutralize the immune system at some close distance, not systemically, but at some close distance, with a high concentration gradient of soluble immune inhibitors. And then as a last-ditch defense, you have membrane immune inhibitors. And cancers just hijack that. It uses the same basic approach and the same molecules, the same specific molecules. And then I would say there's a further, you spoke of hiding from the immune system. Some of the immune inhibitors that cancer secretes, that the placenta secretes, are blinders. So they actually bind and block specific proteins on the surface of immune cells that are detectors, that detect stress proteins. And you can think of that, there's a military analogy for that would be radar jamming. So they're basically jamming the radar of an enemy bomber, right? That's only one class of immune inhibitor. There's also immune inhibitors that shoot down the bomber. So there are molecules that cancer cells secrete and that the placenta secretes, that if an immune cell gets too close, that cell will, they're not generally killed, but could be killed, but they'll definitely be put to sleep. Put into anergy is the term. And that's the second category of immune inhibitor. And then the third category, which is the one we're focused on first, you can think of as a missile and an anti-missile missile. So all immune cells, pretty much, most immune cells, let's say, have one of their main cytotoxic swords is a molecule called TNF alpha. So they'll deliver this molecule focally against an immune cell. It can kill that cell directly, and it also can activate and recruit other immune cells to join the fight. It's a general pro-inflammatory cytokine or signal molecule, okay? What cancer does to defend itself against that incoming attack, in other words, the cells are now irrelevant. The cells have launched this highly potent cytotoxic missile, if you will, against the cancer cell. How does the cancer cell defend itself against an attack that's already underway? Killing or repelling the immune cell at that point wouldn't do the trick. So what it does, and all cancers do this, all solid tumor cancers do this, it cleaves off the extracellular, the portion outside the cell, the receptor, the antenna that receives that death signal from TNF alpha. And that receptor is called a TNFR or TNF receptor. And what cancers do is they cleave it off so that it leaves the cancer cell, but it still maintains binding affinity for incoming TNF. It'll bind it and neutralize it like an antibody. But then the signal, the death signal from that incoming TNF, can no longer be transduced into the cancer cell. And the cancer cell is now protected. It continues to grow and continue to make these TNF receptors and cleave them off and into a very high concentration bubble of protection, a protective shield inside the tumor microenvironment. And that's the target we're going after. These soluble TNF receptors, they're called sTNFRs, for soluble TNF receptors. If you take those out, then immune cells can now deliver TNF and directly against a cancer cell and destroy it. And that's what we've proven works in animals. And also there's a group in Germany that does it by pheresis and has documented the highest efficacy ever recorded against more than 25 different cancer types. So that's our first product that we're going to bring to human clinical studies.
And I think you were in one of your studies or one of the partnerships you have, like you guys tried even increasing the doses of NaNots. I remember not is very important in the name there. 100 times, and it still had no toxicity because they're so small. You could have 1 billion of them in a local area. They really wouldn't bump into each other that much because they're so small. Which leads me to the question: is there any value in making NaNots that each target a different inhibitor and bring it all at the same time? so that you're hitting multiple aspects of an inhibitor sequence? Or I get, that might be like maybe too complicated, considering like most clinical studies or that I've read about or that I've had on the show, they like to make it as like narrowly focused as possible. They can say like, hey, there's this other stuff you could do at the same time, but it's like maybe too complicated. Especially when I think that you guys are working on getting the FDA to see the nanots more as a platform itself versus a drug, so then you could like re-engineer it. But is it possible to administer the nanots, given the fact that you can scale up to 100 times more than what you've done and have no effect, so you can be hitting multiple things at the same time when it comes to cancer?
Well, actually, that's not just a smart question. It's almost like you were eavesdropping on a conversation we had with our team this morning. So there is a debate internally in our group as to whether we're going to make individual nanots against these individual inhibitors and then combine them in one syringe to take out multiple inhibitors at the same time. Or does it make more sense to create one nanot that has capture agents against multiple targets on that nanot? And our team is divided. I feel that well, I'm going to just not say who takes what position, but I'll say
Yeah.
one position in our team is that you maintain combinatorial flexibility by only making one nanot against one target, and then you can combine them in the syringe at multiple. You pick the ones you want, put the concentration in the way you want, and as your disease with different diseases or even different concentrations of target in a given patient, you have flexibility. The other point of view is that from the FDA's point of view, individual nanots are separate products, whereas a single nanot that can take out multiple targets at one time, that's one product. So we can potentially go through the FDA approval process faster with a nanot with multiple capture agents targeting multiple soluble targets than we can taking multiple nanots through the approval process. We need to get more guidance from the FDA. We're working on that to figure out what's the fast track here. I will clarify one point, which is nanots are not non-toxic because well, first of all, we haven't seen any toxicity dosing them at 100 times the planned dose. That doesn't mean there isn't any toxicity, but normally it would have shown up by now with a drug. That level of safety in preclinical studies is really pretty remarkable. We're not actually surprised because, first of all, the materials we're using are safe. Secondly, we're not taking out anything that belongs in the body, at least not at this concentration. So we're taking something out that's tumor generated, that doesn't belong in the body at all. And the third reason we're not surprised by the safety profile so far is this concept of shielding. So nanots are adsorptive particles. They're basically like a sponge. They soak up something from blood. And to do that, they use a capture agent. Now, that capture agent would be bioactive if it could touch a cell surface. We only want it to deplete soluble targets in blood. We don't want it to do anything to cell surfaces because that's where the vast majority of drug side effects come from. So we have a concept which is in our patents, it's patented, and we just call it shielding. And the idea is the capture agents are nested down in the middle of this porous forest of shielding molecules. When the nanot as a whole touches a cell, the cell doesn't see anything except these inert shielding molecules. But the soluble targets diffuse through that shield and get captured by the capture agents on the core of the particle. So it's a really neat geometric, non-chemical, geometric solution to the challenge, this fundamental biological challenge. How do you take out a soluble target that's driving a disease and not disturb the membrane form of that same target that's essential for health?
So a couple follow-up questions there. So for singular targets versus multiple targets on a single nanot, is there any complexity added in manufacturing it or making it, or is it not that big of a difference?
You mean, is there additional complexity to make a nanot that targets more than one thing on that same nanot? Yeah, there's additional complexity. So We use click chemistry, which won the Nobel Prize in 20, actually 2 years ago, won the Nobel Prize. And not we had nothing to do with it, but we've been using it for several years now, because it enables us to attach multiple moieties or molecular structures to the same particle in a very precise balance. It's like a Lego form of chemistry, but there's a limit to how many things you can attach to one particle in a precise way. So we know right now we attach two things. We attach the capture agent and we attach the shielding molecules, those two things. And we have great precision and control to put them in just the right balance. They have to be in the right balance, because if you have more capture agent than shielding, your particle will grab its target very efficiently, but it won't circulate very long because the immune system will see it. Two things will happen. One, the immune system will see the capture agents instead of the shielding, and it'll clear them out. And two, the toxicity you're trying to avoid by engaging membrane proteins will be lost. You'll incur that toxicity. The protection will be lost. So you have to have the right balance of shielding and capture agents. And if you get that balance right, you'll have very high absorptive capacity. You'll have no toxicity because there'll be no engagement of membrane proteins. And you'll have a very stealthy particle that circulates for a long time. Click chemistry allows us to do that when we only have two moieties or structures on the surface of the particle. We can do it up to 3, so we could add 2 capture agents. I think we might be able to go to 3 capture agents, but the process starts to get harder and harder to control during the fabrication itself. So that's probably all I should really say about our internal chemistry. I think there's no limit to how many we can combine in the same syringe because they don't interact with each other. So that approach is open-ended.
Is there so I'm getting stuck on the idea of how it absorbs. So is it attractive? So I think of it kind of like a homeostasis in a membrane, and I think of that as more of an attractive thing because there's a difference of balance, and so it slowly like pulls through to like equal amount. Or is it kind of like it bumps into things, and so normal cells, like you were saying, are ignoring it because it's embedded deeper. But if you're the right cell, it would like, I don't know, like suction in it or something. So how does the absorption happen?
That's a really smart question. And I'll take a little step back and say most nanomedicines have failed. And the reason they've failed is an inherent contradiction or dilemma in the design. So most nanomedicines have been designed to deliver a drug to a tissue, like to deliver a toxic drug to a cancer cell in a certain tissue, right? So they have to do 2 things. It has to circulate long enough. It has to have targeting moieties, targeting molecules that specifically target that cancer cell and not other cells. And it also has to be stealthy enough to circulate long enough for that tissue targeting to be effective. The problem is if your targeting moieties are the outermost element on your particle, the particle isn't stealthy because the immune system will see those targeting moieties and say, that doesn't belong in the body. I'm going to grab it, clear it out. Mm-hmm. On the other hand, if your stealth moieties are outermost on the particle, your particle will be stealthy, but it won't have any tissue targeting intelligence because those moieties are buried under the stealth. And that's an inherent challenge, really impossible to overcome for conventional nanomedical design, and that's why most nanomedicines have failed. They've taken hundreds of millions of dollars of investor money with them. Okay, how do we get around that? Well, first of all, we're not targeting tissue. Our targets are in blood, right? And we believe blood goes everywhere in the body. No tissue can survive without it. So if you want to affect what's going on in tissue, you can modulate what's happening in blood and affect any tissue in the body. So when you inject a dose of NaNots targeting a specific tumor-generated immune inhibitor, for instance, what happens is, okay, we're putting in a very, very low dose in terms of the mass of the nanots, might be a whole dose might be 100 milligrams, so basically the size of a baby aspirin. But it's distributed across trillions of particles. So within a few heartbeats, those particles, the typical nanot and the typical target in blood that we're trying to take out, they're less than a micron apart. Just because a trillion particles distributed through the blood volume, you can do the math here, and they end up, we know the concentration of target, we know the concentration of nanots within a few heartbeats, they're less than a micron apart. So Brownian motion, which is another name for diffusion, causes a random walk of the signal molecules, the targets we're trying to take out. It's the targets that find the nanots. The targets are 1,000,000 times smaller than the nanots. They're vibrating everywhere. They bump into a nanot. The correct target gets grabbed by the nanot. First impact happens in seconds. And the wrong target just bounce off or pass through the nanot. But the right target will get captured. And eventually the nanot will get cleared by macrophage phagocytosis, meaning macrophages engulf them and digest them. And they get cleared with the targets they've captured. But that capture process is blazingly fast. First impact happens within seconds. The nanot dose will deplete its target by greater than 95% at the 5-minute time point. So within 5 minutes, most of that target is gone. It's just been absorbed very, very quickly by the nanots. And then what happens, so your ultimate goal is to affect what's happening in tissue, right? And your initial effect is only in blood. But there's a phenomenon called a diffusion sink. And we're not hypothesizing, this is fact. When you deplete from blood, that creates kind of like a vacuum. It's a deficit of target. It's called a diffusion sink. And that induces migration out from any elevated concentration, any tissue compartment where that target is elevated, because everything in the body is fluidically connected, has to be, or the tissue would die. So you end up with a migration of that target out of the tumor microenvironment, and then you still have circulating nanots in blood. They just continue to absorb that target. And eventually, and pretty quickly, the target of that specific immune inhibitor, tumor-generated immune inhibitor you're aiming to deplete, has been depleted from has been depleted from the tumor microenvironment. And at that point, the tumor is defenseless against the immune system, and you get immune-mediated attack on the tumor. Again, without any drugs or radiation, just by taking something out.
Yeah. So that's really interesting. It's both then. They bump into it. They also create an environment for that homeostasis type gradient to happen. So it gets leached out. That's really cool. So I think this comes to our fan question. Because we keep talking about, getting into people's systems. So I think it's Lexki Moana. I don't know. Moana, you know who you are. What would be the ideal injection administration route and frequency for these types of therapies? I think that was one of the cool things I was reading about in your papers or in the lifespan post, is that you can. Well, I'll let you answer the question, then I have a follow-up question. I won't jump the shark. So yeah, I can reread it. What is the ideal injection? Go ahead, sorry.
Help me interpret the question. Is your fan asking, like, do you have to inject it IV versus under the skin, or?
Yeah, I think they're trying to discern, like, how would it be injected, and then how often to be effective.
Okay, yeah, it's a good question. And it highlights one of the distinctions of what we're doing versus a drug. So in the history of drug design, we're in a new age of rational drug design, but that's not what most drug design has been for the last hundreds of years, certainly the last century. Most drug design has been done by correlation, where you have a molecule you know to be toxic, you throw it into cell culture, see if it'll kill a cancer cell, and if it does, you see if a mouse can survive it, and if it does, you see if a human can survive it and derive benefit from it. And even today, when drugs are administered, even drugs that were developed with a more rational approach rather than just correlation, it's still based on hypothesis stemming from clinical trials, that if you put this drug in at a given dose, in a patient that has this cancer, you're likely to see a benefit. But it's only very rarely with certain drugs that there's anything to measure in the blood or even in the tumor that tells you whether that drug will work. There are some drugs where they know that there's a specific mutation in that specific cancer, and the drug will be particularly effective because of that mutation. But most drugs, there's nothing you can measure in blood that's going to give you any insight into whether what you're going to administer is going to be effective. That's not the case with nanots, because remember, nanots are subtractive. All they do is take something out. Well, that thing they take out pre-exists in the blood. You can measure it. If it isn't present in a given patient, the nanot isn't going to do anything. So the first test, the inclusion criterion for that patient is the tumor-generated immune inhibitor, basically the question is, which tumor-generated immune inhibitors are elevated? Because that's all the nanots are going to do is take those out. And the ones that are elevated are the ones that tumor is using to defend itself, necessarily. Can't survive without them. So, if you pick the main one that's elevated, and you deplete that, and by the way, you can measure that. There's a off-the-shelf ELISAs, blood tests that will quantify that target both before you put nanots in, so it's the right nanot for that target, and know how to dose it. But you can also run the same ELISA 30 minutes later to confirm that you actually depleted the target. And that's very different than drugs. This is called a proximal endpoint, meaning a first endpoint. The first endpoint here is, did the nanot actually do what it was designed to do? Did it deplete the target? And we know, we've tested this repeatedly in rodents, and we know that pretty much every nanot will do what it's engineered to do. It will deplete the target it is engineered to deplete. It'll do it very quickly, within 5 minutes, typically, and very deeply, sometimes down to undetectable levels, but typically more than 90%. 95% is very common. The second endpoint is, do you see a clinical improvement? It's great to have that proximal endpoint. That's very powerful. Then you have to look for the clinical endpoint of improvement. And for that to occur, you have to have chosen your target correctly, and your understanding of the foundational biology has to be solid, that target is in fact a driver of that disease. Fortunately, we don't have to actually do a lot of that foundational science. It's been done for us. There's thousands of published papers by hundreds of teams all over the world on specific soluble targets that are known to drive different diseases or defend tumors. So we're just basically trolling the literature, absorbing all the information, the consensus that's already out there by all these great teams on soluble targets in blood that drive disease, and then we make nanots against those targets.
So you guys are nanots yourselves in some ways. You're
yes, we're scavenging information. That's a good point.
Yes. Yeah. So I was just thinking of the idea of, when your doctor's concerned that you might have a disease or something going on in your body outside of just a routine checkup of taking your blood. They usually just take blood samples. I was just wondering, they could also do maybe an injection and then do like a 5-minute later blood test to see if there's like a certain gradient of like a cancer marker inhibitor in the blood to determine if there's something there. I was just thinking, is there potential to use this type of technology as like a sensor or a way to test if because a lot of the cancer tests out there are not very good. A lot of them are like not great. So I'm just thinking about the potential there.
Well, we actually would agree that the tests aren't very good, and it's mainly because they're not actually measuring everything they should measure. The idea that we know that tumors generate these soluble immune inhibitors, and yet they're not measuring them in a routine diagnostic context. We know what they are. So STNFRs are ubiquitous. You shouldn't have elevated levels of STNFRs unless you have a major inflammatory disease or cancer underway. Excuse me. Good idea having the water ready.
Yeah.
So there's no real reason to have a diagnostic nanot because the targets are measurable. They're measurable with existing off-the-shelf technology. And it's just an ELISA, and increasingly there are bedside devices that are multiplexed ELISAs, so they'll quantify multiple targets with a single blood sample. It's kind of like what Theranos was trying to do, but they didn't. They had their biophysics. They misrepresented the biophysics. The Theranos concept was sound. They just misrepresented the biophysics and lied about their engineering progress. But other companies have successfully made multiplexed ELISAs that can test for multiple targets, not with a single like tiny drop of blood, but with a reasonable blood sample, you can get that quantification of target. So I don't think nanots need to be involved on the diagnostic side. We're developing a way to deplete those targets, an injectable way to deplete targets that drive disease, and not do anything else. Doing only one thing is actually really hard. And we've put a lot of thought into designing nanots to do just one thing, take something out of blood, not engage anything else. And we've succeeded. They will do that.
Just a quick aside on the Theranos point you just made. I read Bad Blood, and it doesn't, in my opinion, so I'm just kind of curious what you think on this subject. Were they ever on a pathway to getting anywhere close to what their mission was? Because from what I understand, they were even like using normal blood sampling technology and like masking it. From what I understand, they weren't even really developing like R&D to like make what they were trying to make, which also was a problem. Like, so do you think if the funding continued and they didn't, start messing with humans at all, because they were kind of freaking people out, do you have cancer? Do you not have cancer? Do you think eventually they would have met it? Do you think they would have like achieved their mission?
I don't know that team ever could have been successful because integrity around data is central to scientific progress. But I will tell you one little interesting anecdote. Our chief technology officer, who's a physicist, PhD in physical chemistry, which is the overlap between physics and chemistry, just what we need, for our actual nanot engineering, actual nanot design. When Theranos first announced their test with their little nano container and all the tests that they would run, our CTO said, that's bullshit. And the reason he knew it was bullshit is that you need when you have a given blood volume and you're running multiple tests on that blood volume, and we have to do this too, you need a certain concentration of target in that blood volume to have statistical validity, statistical significance. I'll give you one example. So say you have a blood volume where you literally only have one molecule of your target. And then you have another blood volume where you have 3 molecules of your target. Well, your test would tend to indicate that sample B had 3 times the concentration of sample A. But in a volume that small, a variation between 1 molecule and 3 molecules is not statistically significant. That could easily be just random that happens. If you had a much larger volume for a molecular species that rare, a molecular target that rare, you would see that the difference between sample A and sample B, let's say the samples are 100 times larger, you would see that you have 1,100 molecules in sample A and 900 molecules in sample B, or probably it'd be more like orders of magnitude more than that. But it could actually be the reverse of what you saw when you only had 1 molecule and 3 molecules present. But it would be statistically more valuable, more reliable, because you have a larger sample and a larger set of molecules. So the variations between them are going to reflect better what's going on in the whole organism. Okay. Well, he just crunched the numbers off the top of his head and said this is bullshit because the number of tests they're doing in a sample size that small, there aren't enough of the target molecules to create statistical validity. So, of course, that's something we have to think about a lot. Anytime we're quantifying a target for treating disease, we need a certain volume size, certain sample size, in order to know what we're doing. Mm-hmm. So I guess to answer your question, a different team that was more honest and understood the physics and was more transparent about the physics could have made a pretty good product that tested. It didn't have to be that number. How many tests were they doing again? I can't remember. It was over 100.
A lot. Yeah, it was a lot.
The systems that are out there that are really reliable, like there's a company called Biotech that has a microwave-sized device called the ELA, E-L-L-A, which is really cool. And it uses a microfluidic chip, and it uses a relatively small blood sample, but it only does 8 tests. And the specific tests that it offers, you can get different chips with different tests, but the specific tests that it offers have been chosen because a blood sample, or can use, I think, urine and saliva as well, a blood sample of the volume they're specifying be used in this chip will actually give a reliable, statistically valid concentration of that target in a given patient. So the biophysics were wrong from the beginning, and she was trying to she had a concept in mind, and she was going to ride roughshod over any objections, unfortunately, including science and physics.
Mm-hmm. Yeah, there's I think it's a TV show that put up on Hulu, and it was pretty wild. Like, I think like she like drove people like suicide and stuff like that. Which everyone looks at people like Steve Jobs and think, oh, the goal there is to be an asshole. It's like, no, I think people like Steve Jobs are the exception to the rule. And I think I even had someone on the show who was saying that there's probably a person underneath Steve who, after Steve was a piece of shit, would go up to them and be like, hey, it's all right, we're gonna be great, just ignore Steve, kind of like that good cop bad cop thing. But I think a lot of people, I think it's better to like treat your employees very well and not drive them to suicide. I don't know, that's maybe controversial, but so in relation to partnering with people, you're a self-learner like myself, and you like to ingest information. You said we were talking previously, you talked about being a generalist. How do you know when it's time to partner with someone who has a deeper, focused knowledge on the subject versus, because there's a level, like personally for me, like when I'm learning something new, I like to get to a certain level where like I can hand it off to someone and I kind of know what they're up to. Like, I don't know it to the level like they could do it, obviously. But then when they tell me about it, I know to like, oh, here's some questions I can ask, or like I can be engaged with it versus them being like, well, widget 3.2 is being done. So I have no idea what widget 3.2 is. I just know like how it influences other things. And so I'm just curious about your process of partnering with other people to build something. And then underneath that is like, how do you maintain a sufficient level of knowledge in fields that you're not a domain expert in, to have, like that discussion with your team about a single target versus multi-target, as an example?
Well, damn, that's a really good question. I think if you're going to work in science, if you're going to innovate in science and medicine, you have to believe in your capacity to understand. First of all, you're tackling something that's just infinitely complex, and you're never going to be able to understand all of it. And nobody you work with is going to be able to understand all of it as well. So you are going to be stronger to partner with people who have devoted far more time than you have available to understand their piece of the puzzle. If you're going to function, though, as a hub for a team, you want to create real power, real potency on a team. Somebody's got to coordinate all of the members of that team. And in order to do that, you need to be able to speak to them and also speak to the other team leaders using their own language at some sufficient level of understanding, of mutual understanding, that you can coordinate all the elements. So there's no shortcut to build a certain level of understanding. And as an autodidact, it's early on, it would be common that your knowledge gets dismissed. It doesn't matter how many thousands of hours I've spent in the literature learning the basics, the fundamentals. Just without an advanced degree, of course, my knowledge is going to get denigrated. And early on, I would have people say things like, well, the math is really complicated. And I would say, well, walk me through it. I've studied some math. Let's talk about the math. And oftentimes, and that's going to be a win-win, because either you'll find, well, they were bullshitting and the math isn't that complicated, or they don't understand it, or they do understand it. And then at the end of a detailed discussion, you might not totally understand it, but you'll certainly understand more. Mm-hmm. So my goal is to believe in my ability to understand complex processes and know when I've gone far enough into that discipline, far enough to be able to coordinate others who have a much deeper understanding. I can't completely punt and just say, oh, we'll go with what Joe says, because Joe might not be right, or Joe might not be considering the needs of the other team leaders, or frankly, the possible orthogonal views of the other team leaders. Smart people, very smart people, often disagree. Somebody's got to arbitrate. Somebody's got to figure out whose position is more right or more likely to be right. A lot of it's just probabilities. We're moving into a space where there's a lot of unknowns. How do we navigate that space? How do we create a new generation of therapeutics that we're pretty sure is going to work in ways that existing therapeutics don't? But we don't have all the answers. We don't know exactly what it can and can't do. We have to actually move into that space of late stage preclinical studies or even human studies before we really get the answers. How do we build consensus among our team and our investors that a given approach is right? And the only way to do that is to hear everybody's point of view and debate it. You have to really debate it back and forth in a data-rich solid hypotheses and solid data. You debate it back and forth, and you find the through line through everyone's ideas that is most likely to be right. No guarantees, but most likely to be right. And it might not be Joe's point of view and Bob's point of view and Jane's point of view. It might be a piece of each. The best plan usually is pulled from multiple people's ideas. And you have to have an intellectual meritocracy. That means everyone is willing to sacrifice their ego. It was my idea. Everybody has to be willing to set that aside and say, you have to be willing to defend your idea when you truly think it's best, intellectually, not emotionally, but intellectually, and surrender a portion of your idea when you think someone else has got the better plan. And if you pick really mature people to work with, they're willing to do that. They might stick to their guns until you convince them. I have people on my team that, it can be really hard to convince them that I think there's another idea that's better. But I don't give up until they've convinced me or I've convinced them. And that's what a true meritocracy is all about. Everybody sticks to their guns, but respects the better idea when it comes along.
Forming a team, making that meritocracy, the intellectual meritocracy. It sounded like one of the things that you definitely are looking for when you ask people questions, when you're going in your deep dives, the ability to explain themselves, to walk through a discussion, are probably characteristics that you look for when you're grabbing people to bring into a team. But is there other things that you look for? Is there a process you go through as you're moving through a space to find the people that you'll then be like, hey, this would be a good person to bring in, or this person to partner with, that type of thing?
So one of the things I've learned, of course, you need people who have specific knowledge, skills, and experience to fill their role on your team. But I've also learned that people need to have a certain level of maturity, emotional intelligence, and compassion for their teammates in order to be a functioning member of the team. There's no one person that's indispensable. You need to have people who make the whole team stronger, and also we all have to be able to take criticism. For instance, I've been in situations where either I or another team member has been correct in their point of view, the comment they're making, but the way they made it was a distraction. It hurt people's feelings. Yeah. I've done it. Other people have done it with me. There was one that happened recently where somebody was making a certain point, and they said, if you had ever cracked a physiology textbook, you would know that. And that they were making was a perfectly valid point. But the whole, if you'd ever cracked a physiology textbook, that's like, eh. Mm-hmm. But we talked it through, and it's like, well, now that you mention it, I haven't cracked a physiology textbook in quite a while. I've been focusing on immunology, but the larger macro physiology, good point. So it was really 2 points. The specific physiologic mechanism that this person was pointing out was worth pointing out. And the fact that I ought to really spend more time reading physiology, not just immunology, that was a good point too. The way it was phrased, that was kind of inflammatory. And we've all done it on the team, but when we call each other on, it's easy to call each other on information if you have data, but when we call each other on communication styles, it's a little more challenging, but mature people can work through that. And if you cross the line, you apologize. Apologies are incredibly powerful. You just own it and you move on.
Is it a gut thing, or is it so I think like there's Elon Musk, when he interviews people, he asks like these weird brain teasers or whatever, which I like he's famous for. I don't remember them off the top of my head. So is it a gut feeling after you're talking to someone for a period of time to know if they're going to have these qualities, or are there questions and things that you ask to try and to bring it out of them?
I'm familiar with Musk's brain teasers, and Google's famous for this too. Like they'll say, how many golf balls would it take to fill a school bus? And the best answer I ever heard to that was, well, hold on a sec, let me Google that. But no, I don't do that. When I'm interviewing people, I tell them what we're building, and I get their thoughts on what we're building. You can tell right away how well people grasp the concept and whether they're able to contribute to the concept. They'll start putting out ideas, asking smart questions, tying it into their prior work, things they've done in the past. The other thing I've found is mature people are not afraid either of criticism or praise. So some people are very comfortable giving criticism. They're not very comfortable giving praise or giving praising an idea or concept or approach. They think it's maybe less sophisticated than to tear something apart. But it's all information. You really need both kinds of information. If you've got a really good idea and you're on the right track, it's very helpful to have that kind of feedback. And likewise, if the concept has some significant drawbacks, those need to be highlighted as well. I'm not sure I answered your question.
Answer my question.
Yeah.
No, you answered my question. It's kind of similar to Matt Scholes in that he'll tell people exactly what his thing is, and he kind of just like, well, that was one of his things for learning as well. He'd tell people his idea, and then all the ways that they said it was wrong, he would then go research it to learn more, which is great.
I do that too.
Yeah.
I have to say, Matt's one of my favorite scientists, and I see him at conferences regularly, and I very much enjoy talking to him because it is a highly efficient exchange of ideas without any posturing or, ego. Mm-hmm. He's superb, and I think that's partly because he comes from outside traditional biology. He's a computer scientist, so and we're both very geared toward cause and effect. Like some people in biology do a lot of hand waving, like this molecule does this thing in the body, nobody knows why. They're really big on correlation, and the underlying assumption is that biology is just too complex to be understood. This is actually a very common perspective in Silicon Valley, where they're building, they want to apply AI to biological problems. And partly that's because they don't know enough biology to actually engage with hypotheses and rational therapeutic design. So they think they'll just throw AI at it. But people like Matt, who are really interested in cause and effect, which I am too, they believe that if there's an effect, there's a cause. Let's dig into what is actually happening at a molecular level that's driving what we're observing. And by the way, if you can't understand it, if you cannot determine the causal link between something you're doing and something you're observing, you probably ought to stay away from that. As an intervention, as a therapeutic domain to innovate within. If you can't understand basic cause and effect, not because you have a problem, but because there just isn't enough known, then it's probably not a good domain to innovate in. And particularly if you have a correlation you've observed, like a drug that has a certain effect, but you have no knowledge of why, that correlation could evaporate overnight and you'll be left with nothing. On the other hand, if you have an understanding of cause and effect and your initial product doesn't work as well as you hoped, you can leverage your understanding to do follow-on innovation and potentially have success. So in our organization, maybe more than others, we really prioritize understanding the mechanism of action of our products and the data we're observing in the literature and in our own studies.
Mm-hmm. So this comes from Lifespan. I think the article, I need to cite my sources better in my notes, actually. That's a note for me in the future. So you said, then I started talking to prospective investors. Among them was Jonathan Milner, founder of Abcam, the world's largest public company focusing on antibodies, and he became our first seed investor. I'm curious, as someone who's an autodidact, how do you establish sufficient credibility in a subject to make someone to encourage them to want to invest in you, to believe that you're the one who can take it to where it needs to go, versus them like, oh, this is a good idea, I'll wait for a PhD or someone with a credential that kind of proves it out. There's multiple level things that like investors look for. One of them is just like, is this the founder who's going to take it and get it where it needs to be? So since you're an autodidact and you don't have those credentials, what do you do to help them believe in you, I guess? Yeah.
Well, I didn't have those credentials at the time. Now that we have, 31 granted patents, and we've shown that the product works, at least in vitro and in vivo in model species, the product is the credential. So it's less of an issue now than it used to be. But definitely when we were talking to our first seed investors like Jonathan, my strategy at the time was just to present myself for interrogation. Like, Jonathan is a PhD in, I think, biochemistry. He's a professor as well. So he just talked to me, and it's easy. It's harder for someone who's not educated to understand what an autodidact understands. But someone who's highly educated, talking to an autodidact who likewise has self-educated to a sufficient level, well, it's readily apparent. It doesn't take that long. You'd start talking. If you're talking to someone who doesn't understand the domain they're in, and you do, very quickly you'll ask a question that they should know the answer to, and they won't. But if you're talking to someone who's really studied their domain, you're not going to drive off the road as often intellectually. They're going to have cogent responses to your questions, and also they're not afraid to say, "I don't know." Once you've built a really solid foundational understanding in your domain, there's no shame in saying, "Yeah, I haven't studied that yet," or "We're still working on that." And likewise, now that we have a team, I am comfortable saying, "Well, I can give you the top level answer to your question, but you really should go talk to Bob or Jane and get the details on that, because they've dug much more deeply into it. Here's the summary." So I think the fundamental message of how to get someone to take you seriously is present yourself. Just say, "Ask me anything. This is my domain. I have studied the hell out of this. Ask me anything." And just the willingness to do that is step one, credibility. Not hiding behind, emails and whatever, but just say, "Here I am. Ask me anything." And then having done your homework so you really can give a cogent answer. That's step two for sure.
Mm-hmm. So I think you're gearing up, or you're in the middle of doing a $70 to $100 million Series B right now. A, how's that going? And then B, is there anything that we as listeners can do to help out in that raise?
Well, that's a really nice question. I think when you start raising money at that level, we're ready. We're ready to work at that level. We have enough. We have enough in vitro and in vivo data that we are ready to progress. I think the bar is higher for us because our therapeutic is orthogonal to conventional approaches. It's not a engineered cell or an ADC or a checkpoint inhibitor. We think it's next generation. It's beyond those things. But because it's new, the bar is higher and it's a little harder to engage investors who are looking for something more conventional. We're in diligence, though, with half a dozen major investors. Some of them are traditional VC, big names, everybody's heard of. But also because we've been funded entirely to date by family funds focused on specific diseases and longevity. A third of our investors to date are longevity focused. But some big family funds like the Lauder family, they have the largest single family commitment to curing cancer. They've invested, I think, in 3 rounds in our company. But because we have our entire history to date is with high net worth individuals and family funds, we have very strong interest going forward in some of the largest family funds that are now looking at covering our Series B. There are specific organizations that would love to see a breakthrough in sepsis, but that's the most expensive combination of nanots that we would ever present. It's well over $100 million to do a toolkit for treating sepsis. So we're progressing toward that incrementally. As far as what people can do to help, I would say we know we're going to close our Series B eventually. I would say it's 50-50. We'll do that within 2 months. It's 80-20. We'll do that within 6 months. It's impossible to know exactly when. As far as how people could help, put the word out. If you're excited by what we're building and the possibilities of this platform, and you're connected to big investors, make a connection.
And the link to contact you will be in the show notes as well. Not your personal email, to your website, which I really enjoyed your website as well. One of the big debates, and I think we touched on this earlier, is this drug versus device. This ability to make something that can be repurposed for so many different things, really a platform, and how FDA sees you guys more as a drug than as a device and a platform. I think that's like one of the struggles you guys are going through. What do you think it would take to get the FDA to see you guys more as like a platform technology? Because I think that would really shift you into a whole new category.
Right.
My understanding. Yeah.
So you have to go with how the FDA views you. When I conceptualize what we're doing, I have a very strong engineering orientation, and I view us as a device. Just as conceptually, I view us as a device with a biologic capture agent at its core. But functionally, it's really device-like. But my view is really irrelevant because what matters is how the FDA views you. And we asked the FDA, are we a drug or are we a device? Because there's different pathways. And the FDA said, you're a drug. It might be different if we were going first to the EMA, the European Medical Authority, rather than the FDA. And it comes down to how they distinguish drug versus device. In the EMA, they say if your therapeutic has a pharmacologic activity, and what that means is that you have a biologic element or entity that's acting on a receptor on a cell or inside a cell, but it's acting on that. That's pharmacologic activity. It's doing something into a cell. That's the EMA definition. We don't do that because our capture agents don't interact with cells. They don't interact with anything in the body except they capture something from blood, but they don't interact with a cell. So it's not really pharmacologic activity. Now, it can have a pharmacologic effect. Depleting that target can change all sorts of ways that cells survive, don't survive, proliferate, don't proliferate. But it's not having a direct pharmacologic effect. But the FDA doesn't use that definition. They substitute the word chemical for pharmacologic. So they say your therapeutic is a drug, not a device, if it has chemical action within the body. Well, we do have chemical action because our capture agents use chemistry to grab their targets. So we can't really win that debate with the FDA. And if we start with the FDA, the rest of the world is pretty easy. They follow the FDA's recommendations, maybe with some additional paperwork required or follow-up studies, but most of the world follows an FDA approval largely. In terms of what it would take for the FDA to treat us as a platform, we think that will happen organically. In other words, we're going to get grilled over that slow rotisserie, the first therapeutic we put through the FDA. There's no question about that. They're going to want to know everything about the platform. But keep in mind, we're using this click chemistry, this Lego chemistry to build the nanot. Most of that doesn't change as we swap out the capture agent to go after a different target to treat a different disease or treat the same disease in a different way. So the FDA, these are smart scientists, and they're going to see that, hey, the last time we came through, you asked us all of these questions. Most of that hasn't changed. Most of the nanot we previously built is the same in this nanot. Only the capture agent has changed. So, of course, they're going to have a bunch of questions about what is this new capture agent? What is the target you're depleting with that? What is the safety of that capture agent specifically? What is the safety of depleting that target? What is the projected efficacy to offset the risk? They'll have some questions, and we'll have to show them safety and efficacy data. But it's not going to be the intensive grilling that we got the first time around when they didn't know anything about a nanot. And I think that process is going to get easier just naturally over time.
Then, so in the article as well, not long after Dolly the sheep was cloned, I was hired to explore the feasibility of cloning the pet dog of a billionaire. I'm curious, working with high net worth individuals, billionaires, and these types of people, have you learned anything from them in particular, or are they just kind of like average Joes with more money?
Well, it depends on how they made their money, really. And I think there are billionaires out there who inherited their money, and then there are billionaires who made their money through the genius of their innovation. Like, no matter what you think about Musk as a political commentator or what he's done with Twitter or whatever, I really don't think it's credible to argue that he's anything other than one of our generation's greatest engineers and manager of engineering processes. You just have to look at Tesla and SpaceX, and you have your answer right there. These things didn't happen by accident, and Tesla had almost nothing on the market when he took over. I don't think they had anything, period, on the market. He took over, and all the subsequent engineering and everything about the charging network and all that stuff is the fruit of his brain and the team he put together. And likewise, SpaceX, the things they've achieved are incredible. So that's a billionaire who is a billionaire for a reason. And the people we've dealt with are a mix of both. So we've dealt with some billionaires who inherited a lot of money and have not really done that much in terms of their personal development, but that's the exception. In most cases, to inherit money is actually quite a burden in how you're viewed. If you inherit a bunch of money, you can be viewed as having no merit, nothing going on upstairs. You just, luck of the draw, you inherited all this money. So it's more common than you'd think that people who inherit a lot of money go out of their way to get advanced degrees or build their understanding, and those are generally the people that we deal with. So there's no one we deal with that, and for one thing, there's some self-selection going on. Someone who isn't smart, like really up to a certain level, isn't going to get their head around what we're doing to the point where they want to put money at risk. I won't say it shouldn't be about intellect. They have to study it. They have to really study it and it's helpful if they're already interested in these things and have studied things like this. Then they're going to get their head around it and they're going to put capital at risk. So in my experience, most of the billionaires we deal with, or high net worth individuals we deal with, by the time they get to us and want to talk about investment, they're pretty sharp. There's a filtering that happens, and the sharp ones are the ones, and well-educated ones are the ones that we engage with.
Did that answer your question? Yes, you did. Well, I think so. What's unique about them? The ones that you're sifting through to you are very sharp and intelligent, and they go out of their way. And like they almost like see a need to do something meaningful, which I think sometimes I think everyone finds meaning in their own way. I guess I don't want to be mean to average people as well. I'm average. I mean, everyone's average in some way. I would just double check and I think you answered my question. I would consider that an answer.
Yeah.
Well, I was thinking that there's like this long debate where it's like, is it intelligence, is it creativity, empathy, etc., that like are important qualities to have when it comes to innovation or engineering of some kind. And there's often Nobel prizes that are the smartest people, the highest IQs, are not the ones who get Nobel prizes. They usually have like an IQ of like 120, 130, which is like above average, but you're not like 160 or something. Is I think how the thing goes. I've been forming a hypothesis that it's actually creativity and empathy that might be like really key quotients to have in yourself to be an innovator. There's this book by Einstein where most of the stuff he did, he just imagined it first. He had the creativity to imagine, a train going by and then, being slow and stuff like that. And then he like kind of intuited and then did the math and everything else underneath it. And I think even what you just said, like it's not so much the intelligence, is like how curious are they, how imaginative are they in terms of like understanding that thing, which I think comes from curiosity. I think that comes from like empathy, because if I think there's a level of like, if you don't empathize with how cool something is, like the impact it could have on someone's life, that type of stuff, you just won't care about it. There's other things you can do with your time. I think empathy, as well, sounds like one of those like key things that's probably separating the high net worth people you've been talking with, not from other people, but just like other things that you've noticed in them as well. That'd be a guess.
I would agree with that. And again, by the time a prospective investor comes to us, whatever their means, usually they want to talk about the innovation itself, and they want to talk about the difference it'll make in the world. That probably is a key distinction between a billionaire and someone who's simply wealthy. Those who have made money far more than they'll ever need oftentimes are more concerned about their legacy than they are about generating further wealth. We talked to some investors who they don't really care about making money at this point. What they care about, they're going to die. Their family members, they're going to get sick and die. Their family members are going to get sick and die. What can they do? That's what's really going to affect them, not running out of money. It's the same challenges of health and wellness and suffering that affect all of us eventually. And that's where they want to apply their resources. And we want that too. I'm comfortable at this point, have worked hard, have some savings. I'm not doing this to become fantastically wealthy. I don't have, what would I spend it on? I'm not motivated by that. I'm motivated. I have a son. I want him to live in a world that has better prospects for health and wellness. And frankly, I'm also motivated by the badass challenge of treating these intractable diseases with huge unmet need. These are really badass challenges. So I love that side of things, like a game. But money just doesn't really. The main issue about money in my world is I can't do the work I want to do without it. We'd already have approved products on the market if we'd never had to worry about funding. I spend 80% of my time dealing with investors, raising money. and 20% actually working with the technology and improving it and writing white papers for scientists, that sort of thing. If we didn't have to think about money, these things would already be on the market. They'd already be approved. So, it will get easier. Once the first one's approved, then everybody throws money at you, or you have access to low-cost capital is the more precise term. That will happen eventually. But for now, it's a struggle. It's a real struggle.
All right. So I know we're coming to the end, so I'm going to send you some rapid-fire questions. If you don't have an answer, you can say pass. What are some books what are some book recommendations that you've gifted to people recently?
Let's see. Book.
I'm always looking for more things to read.
Oh, okay.
As you can tell.
Have you read Seven Eves?
Yes. It's the one where there's like 50 years and then it skips into the future, and it's made from like Seven Moms by Stevenson.
I loved that one. And also Dodge in Hell was great as well. Honestly, I spend most of my time in hard scientific papers, usually by the end of the day, reading a novel. I majored in English literature at Princeton. I read thousands and thousands of pages of literature, and at this point, it doesn't really interest me as much as hard science.
So there's not like a book or genre that you're on the lookout for?
Well, I particularly still love science fiction. So you have a particularly great science fiction novel. I also read history, and it seems like lately it's been Max Hastings' Histories of War, just because a war tends to incorporate so many elements of culture and technology and history all by itself, and geography, and that I've found that to be pretty interesting. But anyway.
Yeah. You might like Old Man's War by John Scalzi. It's like a trilogy, and then there's a bunch of books after it. Basic idea is that when people are 80 years old, they are made young again and made to go fight aliens, which is kind of cool. They get like green skin so they can photosynthesize and stuff. It's kind of neat. But it's great series. I love John Scalzi. And then I'll think of some war books. I know there's like John Keegan's World War II, or the
I've read his stuff. Keegan's stuff I've read. I have a whole shelf of his stuff. He wrote about World War I as well.
Yeah. And then there's like the 3-part series on like Nazis. It's like the rise of the Third Reich, the Third Reich in power, and then like Third Reich at war. And it like really goes into it, like the World War II. Because I'm in America, so I read everything from like an American lens. So it's interesting to see like people like the inside of it, what they did, like how did they come to power and that type of thing, and then how they wield that power and stuff.
If you haven't read Max Hastings' books on World War II, I like his stuff the best. And he's written several of them, so many that his wife said, I would rather live through World War II than have you live with you while you write another book about World War II.
Are you a fan of like Band of Brothers?
Yeah, and I'm also watching the new series. I watched Band of Brothers. I watched Pacific. I'm watching Masters of the Air. Those are great.
Yeah, I'm very excited for Masters of the Air right now. Hopefully it keeps going and in a good direction. Though I assume it's not going to be as hopeful.
Incredible.
Yeah,
the details.
Yeah.
For instance, little things like when it's time to bail out, they flip a switch, which sounds an alarm. I'd never seen that in any, 12 O'Clock High or any of the classic World War II air command movies. You never see that. Little details like that feel so authentic. Or the flare gun.
Mm-hmm. Yeah, I've never seen that. That was wild. I was like, because I always thought like, oh, they just use radio or whatever. But then the German people, the Nazis, would be able to hear that. They'd be like, oh, okay, the bombers are coming. But like a flare is kind of limited in terms of like the scope.
Yep.
How do you think about ensuring you're mentally? This might be our last. Yeah, this is probably our last. This is our last question. Mental health. Is there anything you do to promote your mental health? Like, do you meditate, or just how you think about? It's something that people really don't talk about. So I'm always looking to get people strategies and processes.
I do think about mental health a lot. One of the things I've learned is that I can't work more than a certain number of hours a day. My brain is like a big boat, and it takes a long time to move it from one sea lane, to another. And when I work 6 hours straight, my brain is in a lane that's heading out into the open sea of further work and scientific thinking. And if I want to bring it back to doc so I can sleep, if I don't do that, I will be up all night. My sleep schedule will be completely wrecked. I won't be able to do this kind of Zoom, I won't be able to do any kind of scientific discussion. I won't be able to work effectively if I don't stop working and I have to have my stupid time. I have to have my non-scientific time. And I play a lot of music, both other people's music, and also I work with my son, who's a musician. We record stuff. The point is I do things that aren't science. And then in the evenings, I'll chill with my wife. I go out with my dog a lot. I've learned these are ways to stay healthy. And I've learned the hard way. I try to have every day have islands of peace in the day. And like I said, I do a lot of just sitting and reading the literature to stay on top of it, or writing stuff. But I also take walks. I enjoy quiet time. I used to meditate every day. I don't really do that anymore, but I do respect it. Glass of wine in the evening, once in a while, not every night. It's just important to think about, like, what do I need? And listen to your mind and body as far as what you need, and give it that, support it. Great. It's great to have a loving, significant other, whether it's wife or best friend or husband or mate of some kind, but close human relationships that get you, that are supportive, of whatever you're going through. That's essential.
Yeah. All right, well then, Lou, thanks so
What
much
do you
for coming
do?
on the What do I do? Okay.
Yeah.
Well, we're going over, so I don't know. But what do I do? I have been trying to meditate more. I'm trying to do that. I want to get up to doing it 5 times a day because I'm curious. I've been researching the Quran, and they have like this practice where they pray like 5 times a day. But for them, it's like not so much prayer as it is meditation. So essentially, it's a group of people that basically invented the number zero and algebra and stuff like that. It's like, oh, I wonder what it would be like if I just meditated 5 times a day. So I'm trying to get up to that. I hang out with my wife. That's always fun. I read. I do the walks as well. That's like a big thing for me. And I think a little bit the same thing as you. I try to keep everything like on task. If I tag switch a lot, I feel like I'm not getting enough done, and it bothers me. If I don't get if a day goes by and nothing's good come from it, is in terms of how I define it, it bothers me a lot. So I also like journal, like, hey, this is what I accomplished today, or this is what I was trying to do, and that type of thing. That helps because you can do a lot in a day and not even realize it if you're not thinking about it. So I try to do that as well.
That's great stuff. I was before I got into science, I think I mentioned I was deep into film. And one of the things I did, I had a grant from the National Endowment for the Humanities and to do a documentary about Zen. So I spent a lot of time studying with different Zen teachers. I've done several retreats with Thich Nhat Hanh, and it was a big part of my life for a while. And I think of myself as kind of a bad Buddhist in that when I'm in real turmoil, I'll meditate, but not generally. I don't do it every day as I once did. I think the idea of doing it 5 times a day, if you can pull that off, that's great. Go for it.
Yeah. I think of it like your body kind of tells you if I'm like feeling anxious or whatever. It's like, well, I probably haven't been doing the stuff that takes care of myself, or if I get like overly obsessive. And so I'm very curious. Like I've been doing it, more each day for the last like week, and it's like, oh wow, that's like there's a big distinct shift in terms of how I think. And there was I think it's not Amy Poehler, but the lady from Big Bang Theory. She talks about how like meditation, these types of things, it basically allows you to have like a deeper cup. And so like when stress and stuff comes in, it can like overflow your cup, and that's when you start like being rude or like snapping at people and stuff. If everyone's ever been to like Costco on like a Saturday, there's a lot of people snapping at each other. And so I try not to be mean to others and stuff like that. So I find that the more relaxed I am, the less like people cut me off in traffic and stuff like that bothers me, and then I'm a happier person because I'm not being like, oh, you're so swearing at them and stuff. So little stuff like that. I don't know. It helps. I'm still like learning. There's lots of stuff that I'm learning. So But those are things that seem to be working, like the journaling and a little bit of meditation. I'm just curious, like, what the effects of 5 times a day are as an experiment.
Right. Well, Thich Nhat Hanh used to say that our mind is like all it's like a glass of natural organic apple juice where it's normally cloudy, but if you set it on the counter and just let it sit there, it will the sediment will settle and it will become clear. So his view was you don't really have to do anything other than sit still. Just be still and your mind will settle down and things will become much clearer. And that includes um, cause and effect becomes clearer, like the effect of your words and your actions become clearer when your mind is quiet. So I do some of that settling. Again, I don't do it explicitly through meditation, but I do it through my quieter activities like music and walks with the dog and stuff. And I do find when I don't do that is when the rest of my relationships just aren't quite as satisfying, mutually satisfying.
Yeah. Especially with all the pressure you got going on.
Yeah, I guess I don't really feel it as pressure. I feel it more as uh, excitement. Um, and I just the money issue is challenging. That's definitely we're well past the point where we feel we've proven this to the level where it needs to be tested in humans. We're well past that point, and our current investors feel the same. But it's a big jump to the level of funding needed to achieve that. So it's inevitable, but things can be inevitable and still be frustrating in in the duration required to achieve them.
Yeah.
This has been really fun. You're definitely the most thoughtful interviewer I've spoken to, so I appreciate that.